that I don't want to leave too much time in between the two units. And I think we have enough time, even if I have to stop chemistry for the rest of the semester, pick it back up at the end of October. Anyway, so yeah, this is chapter 7, which is on cellular respiration. Cellular respiration, you can see, is going to be broken down, or going to be the process of breaking down organic compounds into ATP. So whereas before we said photosynthesis was the production of organic compounds from CO2 and using light energy. The purpose of cellular respiration, the overall big picture, is to use these organic compounds that were made during photosynthesis and to get the energy out of them. Okay, that's the main focus. So there's going to be a couple of processes involved depending on whether or not oxygen is present. The first thing that's going to happen is a process called glycolysis. Although I'm going to go over it again, of course, in more detail, but just to give you an overview of the whole process. Whether or not oxygen is present is going to determine what happens after glycolysis. So the first step is glycolysis. Then that basically means to break, which is lysis, and glyco referring to glucose. Okay, that is going to release a little bit of ATP, right? Now, if oxygen is absent, we're going to go with this process, which is called fermentation or anaerobic respiration. Okay, anaerobic meaning without air or without oxygen. But that's not going to produce any more ATP, which you can see by the absence of ATP here. So you might think to yourself, then what's the point? If the whole point of cellular respiration is to produce ATP, right? Big picture. That's the point of cellular respiration. And if you don't have any ATP after fermentation, what's the point? The point is basically to keep glycolysis going, but I'll talk about that a bit later. If oxygen is present, which is most of the time, you're going to go through another process called aerobic respiration. And that's going to produce your the majority of your ATP. Okay, so that's the overall, very zoomed out generalization of respiration. Hold on a sec. Okay. Yeah, everyone's here. Okay. So what I'm going to talk about today is glycolysis and fermentation, or the anaerobic pathways. Okay. Most foods contain usable energy stored in complex organic compounds such as proteins, carbohydrates, and fats. Now, all three of these are made during the process of photosynthesis. It doesn't have to be just carbohydrates. All cells break down organic compounds into simpler molecules, a process that releases energy to power cellular activities. That's the whole point of respiration. Okay, I thought that was a video.
This chemical equation summarizes what happens during cellular respiration. Cells transform glucose and oxygen into carbon dioxide, water, and energy with the help of enzymes. Breathing, eating. The energy we take in is used to make ATP. ATP supplies energy for almost everything your body does.
So I want you to notice that this is the exact opposite, except for, let's see, my screen doesn't match this screen. So let's see where, except for the ATP or energy. Nope. If you go the opposite way, that is the equation for photosynthesis. Okay, so respiration is represented by glucose plus oxygen. And glucose, again, we use as a generality for just like food in general. When it requires oxygen, that's going to be aerobic respiration. Remember, that's the pathway that produces the ATP. We're going to be using enzymes, and then as a byproduct, you produce carbon dioxide and water. Carbon dioxide and water are what's the reactants for photosynthesis. So if I write photosynthesis down here, we start with CO2 and water, and we produce oxygen and glucose. So this is the first like connection. Sorry, my chair, the arms of my chair keep rubbing up against the bottom of the desk and it's making this weird noise. So this is the first link between respiration and photosynthesis. They are literally like opposite. They're like the reverse reaction of each other. And so this is, again, what we call a biochemical pathway, where the products of one reaction are used as the reactants for the next and vice versa. Okay, so that's like, again, one of my biggest points in this chapter, besides teaching you what respiration is also to get you to understand the link between respiration and photosynthesis. There's so many things that they have in common. So respiration is the process by which cells make ATP by breaking down organic molecules. Again, those organic molecules could be carbohydrates, proteins, or fats. Now, again, linking back to photosynthesis and also looking at big picture when it comes to living things in general, you have autotrophs that use photosynthesis to convert light energy into chemical energy. This is in organic compounds. But after that, both autotrophs and heterotrophs have to use respiration to break down those organic compounds to release energy. Okay, so that's how overall both processes are used. So autotrophs, just because they use photosynthesis to make food, doesn't mean that they don't need respiration to get the energy out of that food. That's the whole point. So autotrophs have to do both. That's why plant cells have both chloroplasts and mitochondria. So plants and animals use cellular respiration to make carbon dioxide and water, form organic compounds and oxygen. Chloroplasts are where photosynthesis occurs, and the mitochondria is where cellular respiration occurs. You remember when we talked about mitochondria being the powerhouse of the cell? And that's because it produces ATP. That's the whole point. And if you remember back to the structure of chloroplasts, we talked about these inner membranes allowing a buildup of a concentration gradient. And so we're going to see the same thing with mitochondria. We have a double membraned organelle, and we've got this inner membrane called the cristae, this folded inner membrane, and that's where electron transport is going to occur. Yes, cellular respiration also has electron transport, just like photosynthesis does. So here we have a side-by-side mitochondria versus chloroplast. Again, we've got the transmission electron micrograph that comes from an actual microscope. And then the purple and the green is just an illustrated version of the same thing. So again, just pointing out the fact that you've got these stacks of thylakoids forming an inner membrane and the stroma, so that you can pump protons out of the thylakoid into the stroma through ATP synthase. And again, on the inside of the mitochondria, you've got these inner membranes as well, which are kind of like the mitochondria version of thylakoids, but we call them cristae, or crista for one fold, but cristae for plural. That's because of Latin. You can, for every kind of scientific vocabulary quirk is, you can thank Latin for that, because most of our scientific nomenclature or system of naming comes from Latin. So plants, again, use carbon dioxide and water to produce oxygen and organic compounds, and the products of cellular respiration are the reactants of photosynthesis, and vice versa. The products of photosynthesis the reactants of cellular respiration. It sounds like, I know I keep repeating myself, but it's really important that you understand how everything is connected. Okay. So as I said, starting from organic compounds, the first thing that happens, whether or not oxygen is present, is a process called glycolysis. During glycolysis, your organic compounds, specifically glucose, we use that as an example, is converted into 2 molecules of pyruvic acid. Sometimes you might see it be referred to as pyruvate. It's going to be the same thing. This is going to produce a small amount of ATP, not enough for a full-grown or even a newborn human being. It's just like maybe 1 cell, it would be enough. But it also produces another molecule called NADH, not to be confused with NADPH. And I just always remember the P. It doesn't really stand for photosynthesis, but I remember it as photosynthesis, P for photosynthesis. That's not really what the P stands for. It's probably phosphate or phosphide or something like that. I don't remember the full name of NADPH. But again, as I said, there are very, very close similarities between respiration and photosynthesis, and this would be one of them. So again, glycolysis occurs whether oxygen is present or not. So you can see that here. So you got cellular respiration is another name for aerobic. And then if there's no oxygen present, we call it fermentation, or we can call it anaerobic respiration. But if you see just the term cellular respiration, that's referring to aerobic or with air, with oxygen. But both of them start with glycolysis and produce pyruvic acid and a little bit of ATP. So if oxygen is present, you're going to go through aerobic respiration. You're going to produce carbon dioxide and water. And the number one reason why we're all here is ATP. If there's no oxygen present, you're going to enter the anaerobic pathways. And there are 2 anaerobic pathways that I will teach you about. One is called lactic acid fermentation. The other one, alcoholic fermentation. So this half is going to be the focus for today. So aerobic, aero, this is from Greek, actually, not Latin, of or relating to air, and bios, of course, meaning life, ic making it an adjective. So we're describing respiration here. Glycolysis is anaerobic because it doesn't require, and this is the important part, it does not require the presence of oxygen. Just like the Calvin cycle in photosynthesis does not require there to be light like the light reactions do. But that's why I said I do not like it when the Calvin cycle is called the dark reactions, because it doesn't require it to be dark. It can happen during the daytime as well, as with CAM plants, right? CAM plants do Calvin cycle during the day, open their stomata at night because it's too hot and they'll lose too much water if they have their stomata open during the day. So if we put an in front of a word, it means not or without. If oxygen is present in the cell, the pyruvic acid will be broken down and the NADH is going to be used to make large amounts of ATP. If there's no oxygen present, pyruvic acid can enter other pathways. If you combine glycolysis and the anaerobic pathways, we call that fermentation. A lot of the reactions in cellular respiration are redox reactions. Redox is short for, it's actually oxidation reduction. And I'm guessing they reversed the 2 for redox because oxred is not as cute as redox, I guess. But oxidation reduction. These 2 are reactions that occur in pairs. Oxidation is when something loses electrons, and reduction is when you gain electrons. And there is a, what do you call that? Mnemonic device, if you don't know what that is. Mnemonic device. It's like a way of remembering things. There's 2 that you can use for redox. I like Leo Gur because Leo is a lion, right? And Gur, that's what lions do. That stands for lose electrons, oxidation, gain electrons, reduction. Or you can remember oil rig, which is a thing, right? Let me do it this way. Oil rig, and that is oxidation is loss, reduction is gain. Okay, 2 mnemonic devices you can use to remember the difference between oxidation and reduction. So a lot of the reactions that we're going to talk about in respiration are redox reactions. They're basically, okay, my Bluetooth went boo-booey. Redox reactions, they're basically used to transfer electrons from one thing to another. And that's what a really, really big part of NAD and NADH is for. So you see here at the bottom, all my drawing's going to go away because I lost my connection. We have NAD plus and NADH, exactly the same way in photosynthesis that we went from NADP plus to NADPH, same thing. Okay, so these compounds are being used to basically accept or donate electrons in order to be the other half. Because what I was saying before, redox have to occur together. Oxidation and reduction have to occur together because you cannot lose electrons and just have them float away into the ether. Like that doesn't happen. Just as the same, in the same way, sorry, I've had like 5 hours of sleep last night. My brain does not work today. In the same way that if you have reduction by itself, something gains electrons. Where did those electrons come from? Right? They don't just come out of thin air. So this is why oxidation and reduction have to occur together. If molecule A loses electrons, molecule B has to gain them. Sure. Starting with glycolysis. Glycolysis is where you take a 6-carbon molecule of glucose and you oxidize it, right? Oxidation is loss of electrons. It's oxidized to produce 2 3-carbon molecules of pyruvic acid. Now, if glucose is oxidized, that means something else has to be reduced. And this is where your NAD and NADH is going to come in. What's that weird sound again? Okay. So of course, just like we learned in photosynthesis, they're biochemical pathways. So we're going to have a series of chemical reactions to convert glucose to pyruvic acid. And of course, I'm going to explain it. This occurs in the cytosol. So we're not even inside the mitochondria yet, okay? In the cytosol, outside of the mitochondria, but like inside the cell, this is where glycolysis occurs. And it occurs in 4 main steps. Step 1, 2 phosphate groups are attached to glucose, forming a new 6-carbon compound. This should look awfully familiar, having a 6-carbon compound using ATP to put phosphate at the end. Is kind of similar to what we did when we combined CO2 and RUBP. RUBP was like this, 1, 2, 3, 4, 5, right? And when we combined those two, we did 1, 2, 3, 4, 5, 6, right? And then maybe that's it. Do that one. And that one comes from there. Okay, very, very similar, as I said before. So we have in step 1, we're actually going to use 2 ATP. We take a phosphate from each of those. So we're going to produce 2 ADP, which you can see there at the top. And we put a phosphate on each side of glucose. Step 2. Okay, so we have, so far, this is step 1. Step 2, we're going to split that 6-carbon compound into 2 3-carbon molecules of, drumroll, glyceraldehyde 3-phosphate, G3P. Exactly the same compound that we made in the Calvin cycle. Okay, remember I told you the Calvin cycle, at the end, you actually produce 6 G3P, but only one of them was fixed from your carbon dioxide and that leaves the Calvin cycle to go make glucose, this is it. It's the same exact molecule. And it makes sense if you think about the fact that you put two G3P molecules together to form one molecule of glucose. When you split glucose into two molecules, they are G3P, exactly the same thing that we used to make it. Okay, it's the same molecule. Again, linking respiration and photosynthesis together. Step 3, 2 of these G3P molecules are oxidized and each receives a phosphate group. Again, if something is oxidized, something else has to be reduced. In this case, it's 2 NAD+. You add a hydrogen and you've got 2 hydrogen ions. And in this case, you are reducing it because it's gaining electrons. It's gaining electrons as well as a hydrogen. So that's why NAD is there to accept electrons and hydrogen so that G3P can be reduced. Oxidized. Ugh. So the product of that is a new 3-carbon compound. And like I said, the oxidation of G3P has to be paired with the reduction of something, and that something is a molecule called nicotinamide adenine dinucleotide. No, you do not have to remember that. NAD+ is fine. Okay, so you're reducing NAD+ to NADH. And as I said, NAD+ is similar to NADP+ from the light reactions and photosynthesis in that they're both there to accept electrons during redox reactions, because if something loses electrons, these 2 compounds are there to accept them. And the last step, all 4 phosphate groups that have been added are going to come off, and they're going to be added to 4 molecules of ADP, forming 4 molecules of ATP. What you're left with at the end is 2 molecules of pyruvic acid. You also release a couple of water molecules as well. So the phosphate groups that you added in step 1 and step 3 are removed. That 3-carbon compound at the end is pyruvic acid. So you have used 2 ATP. So we start off with -2 ATP. At the end, we add 4 ATP. So our net gain is only 2 ATP. Okay, even though we make 4 at the end, we initially used 2. So the net gain of ATP at the end of glycolysis is 2 ATP. But we also have 2 molecules of pyruvic acid. So of all of the energy in the glucose, for the moment, we have only released, we've only released 2 ATP. The rest of the energy, and I'm pretty sure there's going to be a question about this somewhere, the rest of the energy from glucose is still contained inside these 2 molecules of pyruvic acid. Okay, now at this point, what happens next is going to depend on whether or not oxygen is present. So, right, I already talked about that.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules.
That's your G3P.
Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
Okay. So the next step without oxygen is called anaerobic respiration or fermentation. Now there's going to be two different types of fermentation. There's going to be different kinds of organisms that are going to use these different pathways. The most important thing to understand about fermentation by itself is that it does not produce any additional ATP beyond what glycolysis produces, right? Glycolysis produces only 2 ATP. Fermentation pathways produce 0. The main point, remember I said at the beginning of the lesson, if it doesn't produce any ATP, and the whole point of respiration is to produce ATP, what's the point of fermentation? Well, if there's no oxygen present, you can't do aerobic respiration anyway. So then if you can't do aerobic respiration, you might as well try to keep glycolysis going by regenerating NAD+. Okay, because remember at the end of, let me just go back here. At the end of glycolysis, you have used 2 NAD and you've produced 2 NAD+. So if you cannot reproduce NAD+, then everything stops, including glycolysis. And then you're producing 0 ATP. And at the end, 2 ATP, at the end, because we've got 2 ATP used, 4 ATP made, 2 ATP is better than 0. And so that is why we go to the fermentation pathways. Again, it's only used under very specific conditions, because without recycling the NAD, glycolysis would use all of the NAD, and then glycolysis would stop as soon as all of it is used up. No more ATP would be made. There are some organisms, like single-celled organisms, that can use just fermentation and glycolysis to survive. And one of those is yeast. And that's when I'm going to talk about alcoholic fermentation. But the 2 types I'm going to talk about, lactic acid fermentation, alcoholic fermentation. We actually undergo lactic acid fermentation under certain conditions, not as a whole organism, but remember this is at a cellular level, and we have over 30 trillion cells in our body. How many is that? That's million, billion, trillion. We have over that many cells in our body. And so not all of them obviously are going to be going through lactic acid fermentation at the same time. So I'll go over all of that. First thing is lactic acid fermentation. This is the one that our muscle cells go through if they are overworked, if you do very strenuous exercise, and your breathing and heart rate isn't enough to keep up with the activity of the muscles, especially maybe like weightlifting, things like that, then some of your muscle cells will start to undergo lactic acid fermentation. So let's look at how this works. Again, starting with glucose, going through glycolysis. In glycolysis, you take NAD+ and you reduce it to NADH and a proton, okay? And you get pyruvic acid. During lactic acid fermentation, pyruvic acid is going to be converted into lactic acid by oxidizing NADH and the proton and reducing pyruvic acid to lactic acid. So pyruvic acid is going to be reduced, and these two guys are going to be oxidized back into NAD+. And this is the whole point, okay? Many stars around the sky. This is the whole point of fermentation, is just to reproduce or recycle NAD+ to keep glycolysis going. Because again, glycolysis produces 2 ATP by itself, and 2 ATP is better than 0 ATP. Okay, the resulting 3-carbon compound after pyruvic acid is reduced is called lactic acid. And this happens through enzyme activity, just like everything else in your body. So you're basically taking these 2 hydrogen atoms away and you're adding it to your pyruvic acid, which turns it into lactic acid, and NAD plus gets produced. And that goes back to glycolysis. All right, so there are microorganisms that use lactic acid fermentation, and we take advantage of the fact that bacteria, this is bacteria, you have, if any of you guys are interested in microbiology, I highly recommend taking at least one class because it's fascinating how many species of things that we cannot see can kill us. I got completely obsessed with it in Bio 101, my first year in university. And then I took, I think, three individual microbiology classes. I took microbiology, advanced microbiology, and medical microbiology. And I still have around here somewhere one of my books of infectious diseases, which is like, oh, it's just, I don't know, maybe I'm weird that way, but I just love the fact that there's something that you can't see that can kill you really quickly. Anyway, there's a lot of bacteria that are quite beneficial to us as well. And if any of you guys like yogurt or cheese or sour cream, anything like that, we use lactic acid fermentation, or the fact that bacteria, there's anaerobic bacteria that use lactic acid fermentation, and we use that to make those things. And I actually happen to make, I make yogurt myself and I make cream cheese myself. Super, super easy. If you ever want to learn how to do it, I can tell you how to do it. It's super easy. You do need an oven though, unless it's like April. If it's like really super hot outside, you can make it outside. One of my most proud moments was making what I call driveway yogurt, because like during the summer, I'll make my mixture and then I'll just stick it in the driveway on a baking tray, and it will just cultivate outside in the heat. Whoop. Oh, I go dropping my Apple Pencil. No. Sorry. Let me pick that up again. Okay. So what happens to milk if you leave it outside of the fridge? And let's say you wait 4 days. So you take the milk out and 4 days later, you're like, oh my gosh, I forgot the milk is out. It ferments, okay? There's bacteria that is in milk and is part of a species called lactobacillus. And there is a sugar in milk called lactose. Do you see a pattern here? Lacto, that means milk. Either Greek or Latin, don't know which one. I would probably go with Latin. And bacillus is an indication of the shape of the bacteria. It's a rod shape, which is like that. You got rod shape, you've got coccus, which is round, and you have spirulina, which is spiral. 3 shapes of bacteria. The more you know. Anyway, so you've got a bacteria in the milk. It's going to use the sugar, lactose, and it's going to go through lactic acid fermentation, and it produces lactic acid. Now, last week I tried for the first time to make cottage cheese. It turned out it looked a lot better than it felt. I don't know if you know, like a lot of how we determine whether or not we like food is the way that it feels in our mouth. And the curds were a little bit too fine for my taste. But the main way that you make cottage cheese is you take milk and you heat it up a little bit, and then you add some kind of acid, whether it's vinegar or lemon juice. And what is lactic acid? Ding, 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 ding. It's an acid. Now, normally the lactic acid isn't being produced in your milk because it's refrigerated. And when you refrigerate it, the cold temperature prevents the bacteria from using the sugar. But if you leave it out and the bacteria start to multiply and grow, then more and more of them are going to be using the lactose in milk and producing lactic acid. And that's why milk ferments. It starts to curdle and smell gross. And so that's how we can take advantage of like the way that you make yogurt basically is I take a starter culture, like usually I'll buy a small pot of store-bought yogurt, and you take, like, depending on the size of your jar, I've got like really big pickle jars that hold probably, I don't know, like a liter, I guess, of liquid maybe. and then you put like a big blob of your store-bought yogurt at the bottom, and then you fill the rest with milk and you mix it up really, really well. And I stick it in the oven at about 42, but yeah, about 40, 42 degrees. And then you just leave it in there for about 8 hours. And at that nice warm temperature, bacteria multiply like crazy. Sorry, someone's calling me. They multiply like crazy, and they produce lactic acid, which then creates this yogurt culture. But it also can occur during strenuous exercise, as I said. If your muscle cells are using oxygen faster than your body can supply the muscle cells with oxygen, then they will switch to lactic acid fermentation. Now, it's not going to have any effect on you immediately, but your, let's say you lift weights for a half an hour, and you're like, whew, and your arms feel like really tired afterwards. Maybe it's leg day, so your legs feel like jelly afterwards. And the next day you wake up and you're like, oh my God, my muscles are so sore. That soreness in your muscles is due to the buildup of lactic acid. And so the cell cytosol is going to be more acidic. And it also, depending on how intense your weightlifting is, the acidity is going to reduce the ability of your muscles to contract. So there will come a point where you physically cannot lift any more weight because of the lactic acid. And that's like if you are like, I don't know, if you guys are like into like super exercise or whatever, but you're going to end up getting cramps. Now, eventually your circulatory system is going to wash out or clean out all of that waste product. The lactic acid is going to go back into the blood. It will go to the liver and be converted back into pyruvic acid, which means the pyruvic acid can be used later when oxygen is present and your muscle cells can go back to doing aerobic respiration. How cool is that? That's why your liver is freaking amazing. So not only is your body adapted to, I think something just got delivered, but anyway, they can leave it at the door. Because now my dog's all like staring at the door, like, who's out there? What am I saying? Right. So not only if there's no oxygen there, your muscle cells have the ability to switch gears and like, okay, well, we're just going to keep glycolysis going until oxygen's here. But like, we're going to convert it to lactic acid temporarily. And then after all of that's done and oxygen's present again, the lactic acid can go back to the liver, get reconverted into pyruvic acid so we can still use it. Nothing is wasted. This is the amazing thing about biology. You'll find out as you keep studying more and more, nothing is wasted that doesn't need to be wasted. So if you have a glucose molecule and you break it down into 2 pyruvic acid molecules and there's no oxygen there, your body's just like, man, we're going to throw it out, whatever, we don't need it. It saves it and converts it to lactic acid, changes it back to pyruvic acid so you can use it later. Okay, and then the last thing is alcoholic fermentation. My favorite, just kidding. Okay, hold on a sec. Okay, leaving it at the guard, even better. So again, starting from glucose, you go through glycolysis, you reduce NAD to NADH, and you get pyruvic acid. Same thing. But for alcoholic fermentation, we're going to take a little bit of a turn here. And I'm so sad that we're not in school, because this is the time of the year where you guys get to make wine. Yes, that's right, wine. And it's usually a really fun experiment to do when you can actually see alcoholic fermentation working, but well, COVID. Anyway, you can see from the diagram that during alcoholic fermentation, pyruvic acid is going to be converted into ethyl alcohol. This is also called ethanol. Not the same as methanol, which is the chemical in the lab at school that we put in the alcohol lamps. Although when some people, when people try to distill certain types of ethanol, because ethanol has 2 carbons, methanol has only 1 carbon. Sometimes methanol can be produced. And that's why they say if you try to make moonshine, I don't know what they make moonshine from, potatoes, I think, something like that. You can end up making methanol instead of ethanol. And methanol attacks your optic nerve, which is the nerve that comes straight out of your eyeball and goes to your brain. And so it can cause blindness. So I don't recommend trying to brew your own moonshine. But anyway, you can see that a carbon dioxide is produced. So we're going to take pyruvic acid, which is a 3-carbon compound. We're going to release one of those carbons as CO2. So we have a 2-carbon compound. That 2-carbon compound is going to be reduced, so that NADH and an H can be oxidized. Again, the main point being we want to reproduce NAD+, and ethanol or ethyl alcohol is the result. And where we use this is obviously making alcohol, but also making bread. Okay, so we're going to remove a CO2 from pyruvic acid. We're going to have a 2-carbon compound left over. That's here. And then we have NADH and the hydrogen ion, which are oxidized to NAD+, which means this guy is reduced. So we're taking these 2 hydrogens, we're putting it on there, and that's ethyl alcohol. Again, the whole thing is for glycolysis to keep going. So the obvious uses of alcoholic fermentation is the basis of wine in the beer industry. Also, your hard alcohols like your whiskey, your vodka, gin, blah, blah, blah. The kind of drinking alcohol that you make just depends on what ingredients you start with, what the source of your sugar is. And the organism that does this process is the yeast. Yeast is a type of fungi that cannot produce their own food. They are heterotrophs. Okay, they don't use photosynthesis. They need to use another type of process to get energy, and that is fermentation. If the yeast is supplied with a food that contains sugar, it's going to perform alcoholic fermentation and release ethyl alcohol and CO2. I am sure that you guys at some point in your life have seen yeast being activated. When you buy yeast in the store, you buy it in a jar and it's just like these little granules, right? When you put them in hot water and you add a bit of sugar, eventually it becomes this frothy kind of mess. And those bubbles are CO2. Okay, so this is the setup, and I actually freaking bought these things during the summer to go into this year of biology so that we could actually make wine properly, because I was using like freaking balloons that would get bigger because CO2 is being released. And of course, if you're making wine, you don't want bubbles in it unless it's champagne. So table wine is basically uncarbonated, just like your regular wine. That's what they call it, table wine. And the CO2 is allowed to escape, and that's what this thing is for. So basically you've got this stopper here, and you've got your, I forget what they call this, but I've got 2 different types that I bought. But when the CO2 gets too much here, it pushes this outside thing up just enough so that the air escapes, and then it comes back down to keep it an anaerobic system. If you want to make sparkling wine or beer, you're going to completely seal it off so that you keep the carbon dioxide in it. One of my roommates in college for her chemistry class made beer, and it stinks. That's all I remember from that. There was this big giant keg in our apartment and it stank. Now, we also use alcoholic fermentation to help make bread. The difference is our bread is an alcoholic because alcohol is very volatile. Volatile means it evaporates quickly. And of course, we bake bread, but I don't know if any of you guys have ever tried making bread. My husband's really, really good at making bread. He makes it all the time. And so when you mix, you've got the flour, you've got sugar, you've got your yeast, and a bit of warm water to activate the yeast, and you mix that together, and it almost always involves a rising period. So you generally, and that's what you see here, is that you knead the dough to release the gluten and to make it like stretchy, depending on what the consistency of the bread you want. And then you let it sit. That's called rising. And then because of the CO2, you see that the dough gets really, really puffy. And then you bake it, and that's where you get these holes. That's from the CO2. And of course, it evaporates when you put it in the oven because it's alcohol. Alcohol evaporates quickly. All right. So the last thing before I go, I'm like out of time, but it's okay. I'm gonna take me 3 minutes. How efficient is this? Okay, how we calculate this is a standard amount of glucose contains 686 kilocalories of energy. The standard amount of ATP from ADP absorbs 7 kilocalories. So the energy required to make ATP divided by the energy released by glucose times 100% is going to give us our energy yield. So during the anaerobic processes, we have made a total of 2 ATP molecules. Each one can release 7 kilocalories, and one standard amount of glucose contains 686 kilocalories. Times 100% gives us a grand total of 2% efficiency for glycolysis and fermentation pathways. So of all of the energy in glucose, we only get 2% of the energy out. I'm going to stop there, and I will follow up the rest of this on Friday.